Okay. Righto. So let me share the link with you. Hang on, tick. Here we go. Is that the correct one? Yes. Just going to put the paper in here and on here. Great. And I'm going to share my screen with you. Well, welcome to Neuroccino. It's a small group this week, given that we have Brain Hack in Bordeaux and a bank holiday in the UK for the coronation. So it's just going to be a smaller round. And I got a paper that came out last week, I think it was, and it's quite exciting because it's a tour de force of methods and scope of the paper. There's a lot of clinical potential, which I think is fantastic. But also the ideas behind it are quite intriguing. So the paper is called Semantic Reconstruction of Continuous Language from Non-Invasive Brain Recordings. If we deconstruct that title a little bit, semantic just means meaning, association between words. It actually doesn't reconstruct language, it reconstructs word sequences. So it trained a model to recognize the likelihood of words co-occurring. So it's a classical ChatGPT basically for fMRI data. And the non-invasive brain recordings is a fancy way of saying functional imaging. So there you go, that's the title. Now, what's interesting about this paper is that it's actually linked to a previous paper that came out in 2016, which also has the nickname The Brain Dictionary, where participants were listening to a podcast and they just observed where activations popped up across the brain. And we got a map that looked like this. So you had activations everywhere in the brain linked to semantic knowledge, but semantic just being the concepts of words, not nitty-gritty detailed semantics. So that's an important thing to remember. So what this study basically produced was a colorful quilt that was laid over the cortex. And this atlas then displayed in rainbow colors how individual words and concepts are grouped together into lumps of gray matter, basically. And one of the interesting findings, a couple of interesting findings from this study was that there is no one single brain region that holds all the words or concepts, which is nothing new. But I also showed that it's a lot more distributed than the classical language network that we consider. So coming back to this new study, they now again looked at these high-level semantic features of word sequences in a similar design. So again, the Moth podcast. But what they wanted to get to is basically a brain-computer interface where the computer that was trained on some machine learning, we're going to come to that in a minute, can decode language in the brain. And this is obviously extremely important when we look at clinical settings where patients may not be able to generate output anymore, like articulate. But if you could read their thoughts, basically, then you could still engage in some kind of conversation. Now, the reason they chose semantics in this setting is because you could also do it with motor, but especially in clinical settings, the motor system may not be intact anymore, whereas the conceptual system may still be there. And also previous papers looked at the articulation side of it using brain-computer interfaces. But usually they were quite invasive. So here the beauty is that it's non-invasive. It's based on functional imaging. Now, the biggest obstacle they said the study had was the low temporal resolution of MRI or fMRI, because the bold signal takes about 10 seconds. So you have the delay, and also the rise and fall of the bold is about 10 seconds. Now, if we look at English, which is the only language they tested in the study, we speak with 2+ words per second. And that means that one brain image has about 20+ words encoded in the image. So what they then did was to train the model to generate target words or candidate words, or sequences, I should say, it's not individual words, and selected the most likely sequence to decode the functional imaging. Now, I said it's a tour de force of scope and methods, and when I read it, I identified about 8 individual parts of the paper. So the first one, let me just go to a nice figure so you actually have something nice to see here. So the first one is that they trained their machine learning algorithm across the brain. And what they used as a generative neural network language model, so GPT, and had people listen to over 16 hours of spoken stories. So again, the podcast idea. And they claim that that gives you 5 times more data than a normal experiment, language experiment. They then used a model based on the semantic features of words, and then a simple linear regression to model the brain responses. And the idea is that the encoder scores the likelihood of the match between the predicted and the recorded response. And if it's a good match, then it basically decoded the brain signal. Now, they used a beam search algorithm. No expert in what that means. I didn't have the time to dig deeper, but apparently that made searching for the most likely word sequences a lot better. And then they also used a BERT score to quantify shared word meaning. Now, for a sanity check, what they did was a behavioral experiment, and basically all the word sequences that were reconstructed from the imaging data, they then gave to participants to read the decoded words and check if the gist is there and if that actually is a good match, which seemed to work. So this was the first part of training the algorithm. Then the second part was to bring it back to the brain and find the areas in the brain that are most likely associated with those word sequences. And the brain is generally active during language processing, even though they never really specify what they mean by language or processing in this context. But the idea was to then partition the brain or parcellate the brain to look at the multiple cortical regions that represent language at the granularity of phrases. Now, they did this for the whole brain, but they also wanted to zoom into individual regions. And we can just go to the second figure here so you can, see the cortical areas a bit better. But basically, what they wanted to do is look at the individual regions and their pattern. Now, what they found was that between 25 to 95% of the time points in the time series of the fMRI could be decoded from the brain. from the speech network. So there seems to be quite a good overlap between the individual regions and the signal they're picking up. But what is not clear is if there is a timely pattern in those regions, meaning that the region may be implicated, but they couldn't detect if it was activated for the entire time or for a specific reason. Now, being able to record and predict between individual brain regions and the brain and vice versa is obviously relevant for a clinical setting again, because in a clinical setting, not all brain areas may be accessible or intact. So if you can use other information to then reconstruct, that is obviously helpful. Another question, and this is only part 4 now that I found in the paper, was that idea between modularity and integration. So the idea being that if you decode from multiple regions, the question then is if these different regions encode complementary representations, meaning that different parts of speech are encoded in a modular organization. Now, if that was true, then this would mean that by decoding individual regions, you could decode aspects of speech, but not the full stimulus, right? Now, alternatively, the idea could be that different regions may encode redundant representations of the full stimulus. And in this case, you could reconstruct the same information by decoding different individual brain regions. And this is what the data suggested. So it seems that there is more redundancy than specificity in brain areas. And again, this is clinically relevant because if patients can't produce covert speech, you might be able to reconstruct the gist, at least, from different sources. Now, what they also wanted to look at is, with a clinical perspective, is that not every patient has the ability to express themselves. So what if you don't have access to spoken speech, for example, and you just have imagined speech that you want to decode? So what they did is they asked a couple of participants to go in the scanner and tell 1-minute stories 5 times, and then also repeat those stories outside the scanner for reference transcripts. And then we're checking if the algorithm can detect the most likely transcript from the brain signal, and apparently is 100% accurate. But that's just to pick out the probability of the most likely transcript. And then overall, the analysis showed that it's less accurate in the predictions than perceived speech, so listening to the podcast. But this could be quite exciting to look at in clinical settings going forward. Now, another thing they tested was modality specificity. So they made, again, a couple of people watch silent movies and then compared the brain signature of a silent movie with the auditory description that is available for people with visual impairments. And again, found a good match between the auditory description and the signal they could detect. They were also interested in the influence of attention, because obviously our language system doesn't work in isolation, it works in an embedded cognitive fabric. So they then asked people to, they had overlapping stimuli, and they asked them to pay attention to one or the other stimulus. In that case, they had female and male speakers and overlaid their traces, and then asked them to either attend to the female or the male narrator. And they were able to show in the paper that attention is important and that we can basically decode what we're paying attention to. Now, another thing that was raised and I think is very important and often underrepresented in our literature is the idea of mental privacy or neuroethics. So the idea here being is that obviously this, if you publish a study like this, it's going to be picked up by the media as well. We can read your thoughts, which is definitely not the case. And they showed that people basically had to report and consent to have the brain signature read out, not in the sense that they had to write, well, they also had to give their written permission, but also you can't decode it unless you get the right input. So the model is only as good as the training data that you put in. So there is a limited scope of what you can actually decode. You can't just put a hat on someone and say, oh, I can read your thoughts now. So the compliance during training and readout was shown to be important, but I love that they dedicated an entire section to this in the paper, because that is an important thing, especially when you consider the clinical setting where people may not actually be able to consent properly before you test the method on them. Now, that's all the exciting stuff. The limitations is that they tested a small number of people. So it was 3-4 males, females, I think it was. They're a bit inconsistent across the paper between speech and language. It seems to be used interchangeably. Again, the concept of semantics isn't elaborated on, at least not in the main paper. I didn't read the supplementary yet. So in terms of the granularity that we can get to, it has similar limitations to the 2016 study. And at the moment, there is a lack of generalizability because obviously it was trained in English only. But that's a training matter, I guess. So that is not a biggie. There you go. That is all I had for you. Do you have any questions? Lilith's hand is up. Here we go.
Thank you for presentation. Cool paper. I have a question about, so the participants were healthy, right?
Mm-hmm.
So how are they going to transpose this to clinical population? Because if the data was trained on healthy participants, it's one type of brain with one type of activity. And when you have a stroke, for example, everything will be changed a bit at least. And then the prediction will be, for sure, I think, really low. So how is this going to work with the patients if anyways they need the training data at some point?
A very, very good question. It's not that well specified, again, at least in the main part of the paper. A lot of the clinical links that I mentioned came from me, not from the paper. Except that they mention that it's going to be helpful for clinical populations where you need to recover covert imagined speech. But how exactly that's going to happen and also how that will be implemented or translated into the clinic is specified in the paper. But I guess as long as you have functional data, you can train your algorithm on it. Now, one thing that I was missing was also, let alone clinical populations, which introduce another level of variability in your data based on stroke lesions, so actual areas damaged or missing, degenerative processes, etc., you name it. The general variability in the brain signal in the areas was not reported in the main part of the paper.
Yeah, and also another question is, so there was one of the tasks when they had to actually tell a story in the scanner, right? So here it's not about imagination of a story, but actually articulating it. It's also, I think, different from what if we use this type of method with patients, they will think about something to say, but not articulate. So maybe it would have been better, I don't know, if they actually not ask participants to actually tell a story, but imagine the same story and repeat it in the head, kind of, in mind.
Again, good point. They had two conditions. The first one was the imagined stories, which was basically also to check how generalizable their predictions are, because those were new stories that the participants came up with. But then they also had the silent movie part, where you had the same visual input for people, but they narrated it in their head differently.
Yeah, but it's perception, kind of. It's not when you are actually producing a thought.
Correct.
So it's a bit
correct. But they said that you need to have the training data to decode it. So in that case, it's a circular system, and the test stories had 1,839 words that were then sequences. So if we look at the calculation from the beginning, you would have per image 20 plus words. The entire test story had just shy of 2,000 words. So there's only so much variation in the data that you can play with, which obviously increases your predictability, right? Because you have a narrow set of word sequences that you can actually decode. So if you open it up and have a much larger pool of possible combinations, I'm expecting it to get harder and harder for the model.
But then I have another question. For example, there is a patient, right? Again, sometimes it's really not possible to put a patient in scanner. So if, for example, if everything will work perfectly, if they will be able to actually decode from fMRI scans of whatever patient is thinking, will it be still used? Because it's kind of still, I think it would have been better if it was like EEG or something, which is maybe easier, but still there might be some symptoms that will not allow to pick up the signal perfectly. So I don't know, even if this goal is reached, will it be actually used? Because it's a bit complicated, I think, with patients. What do you think?
I agree. I think it's complicated, and that's why I preempted it with exciting ideas. Also, like the idea of a brain-computer interface, at least the way I understand it, is that you basically plug yourself in, you get feedback, and it's all quite dynamic. Whereas here it's fMRI data that is then analyzed.
Yeah.
Which is the normal way we do things. But obviously talking about a brain-computer interface kind of implies that this can be sped up and be a lot more dynamic and accessible, because an MRI machine is not a brain-computer interface. It's a clunky machine that not many people actually have access to, whereas brain-computer interfaces tend to be like wearables, right? Or at least more portable or more accessible than MRI scan. And especially with patients, we've seen it with stroke patients, for example, that fMRI tasks, especially for patients where language is impaired, fMRI tasks are very difficult to do because you have to tailor the test to the capability of the patient. You can't ask them to do things they can't do in the scanner, because then you don't get reliable results. So one of the great discoveries over the last decades was resting state. So you can scan a patient without needing them to actively participate in the task. So this would be another interesting way to see what you can decode. But then obviously with resting state, we it's easier to double check what we're looking at with fMRI because there's a conceptual framework behind the task. Whereas with resting state, it's a bit more open what we end up seeing.
Okay, thank you.
Sabrina.
Thank you. I think so that with the patients is really difficult. And maybe at the end we should have more data, resting state, as you said, but also from other methods. And but then, of course, should be really nice if at some point we can arrive to do something like this, and maybe this is a good start. Because also another aspect that or question that there is also about help patients in coma. So this could be also a way to help this. Then, of course, there is the other aspect about the consent to do this if someone is in coma, of course, but could be at some point in case a help.
But that's the idea, right? That wherever people become unable to communicate, you find ways to circumvent that. So that could be locked in patients, that could be coma patients. The idea is that you maintain communication with people who can't articulate.
Yeah. And then also, I was wondering about another aspect, if we know from the paper, if we know the brain areas where the output was the same. I mean, where maybe we had the same word or the same output in the brain. I mean, for example, I don't know, maybe in 2 areas or more, we have the same output from this method. And it's interesting to see where we have the same output and the same prediction.
Yeah, I agree. So what they did to begin with was to run a localizer, a speech localizer. And what you can see in the figure that's currently up is that you have the classical speech areas, then you have association cortices in the temporal parietal areas and the prefrontal cortex. So if you compare that to the 2016 paper, for example, where pretty much the entire brain came up, there is a slight difference here, but also some overlap. So yeah.
Okay. This is. Thanks. I see there is Eva.
So distracted by the advertisement. Clearly Google knows that I like Italian sports car and I can only afford the Playmobil version of it. Sorry, Eva, you had a question.
Yeah, just a quick one following up from what Lilith said. Do they mention why they use fMRI then? Because they have the temporal resolution issue. I don't know if it was for the spatial resolution or there were other reasons why they chose fMRI.
Can't remember reading that specifically. I know there's other studies looking at EEG, MEG, and they mention in the paper that we should look at these modalities. I think it was somewhere in the limitations. Let me see if I can. Here we go. This could make use of complementary recording methods such as EEG, MEG, which capture precise timing information that is not captured by fMRI. So future outlook, but that's as far as it goes for the time being, at least in the main manuscript. As I said, I didn't read the supplementary yet. Cool. Any other questions? No. Great. Well, thanks for coming this morning. Thanks for your question. Happy to discuss that in the lab a bit more if you like. Have an exciting week in science. For those of you joining Brain Hack this week in Bordeaux, have a fantastic time and good luck with the Stroke Cup challenge of predicting. For the others, I see you in the lab and see you all next Monday morning for Neuroccino. Bye.
Bye.